So this morning, I'm very excited to welcome our first two speakers. They are gonna each speak, but for interest of time, I'm gonna introduce them at the same time so that we can keep moving. As I was told this morning, we've really packed this agenda. I appreciate everybody hanging in with us through technical issues and not enough breaks and maybe not enough coffee. You've had a great attitude and we really appreciate everyone. So first we have Dr. Alicia Gomes. Dr. Gomes is a licensed and certified genetic counselor. She has served as a pediatric genetic counselor and a molecular laboratory genetic counselor since 2010, and then as lead genetic counselor since 2014. Dr. Gomes' expertise includes molecular genetics techniques and specific focuses on the neurofibromatosis genetic disorders. She also serves as coordinator for the NF variant Curation expert panel, which she'll be talking to us about this morning, and secretary of the Alabama Board of Genetic Counseling. Then also, after Dr. Gomes, we have Dr. Kavita Sarin. Dr. Sarin is an associate professor of dermatology at Stanford University. Her lab has an academic interest in employing molecular and genomic analyses to elucidate the development, progression, and treatment of cutaneous diseases. Dr. Sarin has published over 50 original publications in the area of her research, has been featured in mainstream media outlets as well. She serves as a co-chair for RAIN's Cutaneous Neurofibroma Working Group, which we heard about yesterday, and is also a scientific advisor for the Skin Science Foundation and Inflection Therapeutics. Dr. Sarin also sees patients in medical dermatology and directs a specialized skin cancer genetics clinic at Stanford Cancer Institute. Welcome, Dr. Gomes and Dr. Sarin.
Good morning, everybody. So my name is Alicia Gomes. I am a genetic counselor at UEB, and today I wanted to talk to everyone about the genetics of NF. To do so, I think it's very important that we're all coming from our background of genetics from different places. So I like to start from the very beginning, if that's okay. So we're going to talk about just the basics of genetics in general first. So we all have genetic information in every cell in our body, and it's all bound into packages called chromosomes. So if you unwind those packages, that's where you get the letters or the strands of genetic information, and that's where we have our genes. So there's a very specific alphabet for genes, and it's made of 4 letters, A, T, C, and G. And they have partners, and A always likes to bind with T, and C always likes to bind with G. And so for each gene in the body, there's a specific order in which those letters need to present. So as geneticists, genetic counselors, anyone in the genetics profession, what we're trying to do is read those letters to make sure that they're in the right order. To do so, there are 2 different approaches that we can take. And so to talk about genes, I always like to bring it to a book because that's the easiest analogy that I can think of. With a book, you can have 2 ways to edit it. You can look at each letter and all of the words, or you can look at the paragraphs. The first approach that we always like to take is looking at each of the letters. And there's 2 different methods that we can use to do that. One is called Sanger sequencing. And so if anyone has had testing maybe a decade ago or earlier, Sanger sequencing was the only option that was available. More recently, we have a new technology available called next generation sequencing. But for both, they look at each letter individually to make sure that they're reading in the right order. And if you see a letter that is in a place that it should not be, it gets flagged. So if you had testing, some time ago and it was Sanger sequencing and a gene change was found, there's no need to repeat testing with next generation sequencing. They're both equally as robust. And even with next generation sequencing, we still have to rely on Sanger every now and then. So while each technology has its pros and cons, they're both very valuable. So the types of gene changes, so from here out, I'll call them variants. The types of variants that you can pick up with this technology can be things as simple as switching out a letter, and we call that base pair substitution. So in my example here, if you had a sentence that said the car was gray, and you change that R to a B, then the sentence no longer makes sense. It's the same is true for your genetics. If you change out one letter sometimes, it makes a protein that makes no sense, so the body can't use it. And so that can be a disease-causing gene change. You can also have letters that are either just 1 or 2 or 3 maybe, that are added in or deleted out. And so we call these deletions or duplications. And so this sequencing technology can pick up small deletions and duplications as well. So what's important to remember, though, is that all of us have gene changes and variants, and we all have tens of thousands of them, but that does not mean that we have tens of thousands of genetic disorders. These gene changes are sometimes benign and just make us who we are. And then when they're in specific areas that are concerning for disease, that's when they can be considered what we call pathogenic and disease-causing. So part of what geneticists do and folks that are working in laboratory and research is that they look at these gene changes and they use a set of rules that was developed in 2015 and try to give points to these changes and variants to say, okay, is it more likely that this is a disease-causing gene change, or is it more likely that this is a benign gene change? And so with the point system, it's very universal. It's meant to apply to any gene. So there's a lot of personal interpretation that takes place from lab to lab. But with the points, we try to get to pathogenic if we can, or benign if we can. This isn't always possible, and sometimes we get some points, but just not enough to make it all the way to pathogenic. And so we call those gene changes likely pathogenic. It means that we have about a 90% confidence that we've gotten it right. But when we can't get there, sometimes we just have to put it in a bin called variant of uncertain significance. So I'll leave that there for now because we're going to come back to that point towards the end. There are also different ways that you can have sequencing. For the conditions that we've been talking about, NF and schwannomatosis, in many cases, there's enough features that we're really only concerned about one gene. And when that's the case, you only need testing for one gene. Sometimes you can have a clinical presentation that does require more than one gene to be tested. And so we call those targeted panels. So it's looking at a very select number of genes and doing a good deep read of testing for just those genes. For other individuals, and it's usually more common for other disorders, we can't just make a small list of genes that we're concerned about. And so it may require whole exome or whole genome testing. And this is where we have to test as much genetic material as we are able to at this point to try to see if we can find a disease-causing gene change. But when we're talking in the focus and then under the lens of neurofibromatosis and schwannomatosis, it's usually those first two categories that we find ourselves in. So we talked a lot about sequencing. There's other technology. So if we go back to our book analogy, so we've been looking at each letter individually. There's also the scenario that we need to make sure all the paragraphs are there. So are there any extra paragraphs? Are any of our paragraphs deleted? And so that we do the same thing with our genetic material. Sometimes there are bigger pieces that can be extra or missing. And so we need technology called microarray or MLPA that are utilized to try to figure that out. So I won't go into a lot of detail about it, but I mentioned this just to say that for anyone who's had genetic testing, it's important to make sure that it's included both processes so that you've got a thorough review of all the types of gene changes that could have been picked up. So that's my genetics in a nutshell. We're going to move now to talk more specifically about the neurofibromatoses and schwannomatosis related disorders. So everyone who gets a diagnosis of NF has usually done so through one of two processes. You can get a clinical diagnosis, and this has been the case for many individuals because you have features that are very hallmark for a specific condition. And so in the clinic visit, they're able to say, I know you have NF1. And this is through a specific set of criteria called the NIH criteria, and having 2 or more features lets us know that this is probably correct. Some people don't have enough features to make that clinical diagnosis, and in that case, they may need genetic testing, or genetic testing may be needed because their clinical presentation is a little bit more ambiguous. And so when you have to have genetic testing to confirm, we call that a genetic diagnosis. Recently, in 2021, there was a move in revising the diagnostic criteria for NF1. And there were a lot of reasons for that. One is that there's been some advancements in drug therapies for NF1, and so there's been a need to prove that there's an NF1 mutation in some individuals. Small children especially don't necessarily present with enough features up front that one can feel confident in making a diagnosis off of clinical features alone. So there was the inclusion of genetic testing into the diagnostic criteria so that with cafe au lait spots and a pathogenic NF1 mutation, for example, then you're able to make that diagnosis sooner instead of having to wait. So let's talk a little bit more about NF1 specifically. So it is caused by the NF1 gene located on chromosome 17. It's autosomal dominant, which means that autosomal, it is not on one of the sex cells, so it's not on X or Y, it's on chromosome 17. And dominant simply means that you get one copy of every gene from your mom and one copy of every gene from your dad. And it takes receiving a copy from either mom or dad, just one copy, and you will have NF1. It's what we call a fully penetrant disorder. So fully penetrant means that if you have that gene change at some point before the age of 8 for NF1, you will have at least one feature of this condition. And so those are some important pieces that are unique to NF1. 50% of the time, this condition is caused sporadically, meaning if you look back in multiple generations, you won't see anyone with features of NF1, nor will you see a gene change in NF1. It started brand new in that specific individual. So mosaicism simply just means that only a percentage of the cells are involved. We call it generalized mosaic NF1 when it happens earlier in the embryo development, so there's more area that could potentially be affected. And so on the outside, we may see just a very mildly presenting individual, but there are features kind of all over the body. If it happens later, then it could be called segmental NF because the cells have developed and decided that I'm only going to be arm cells now at this point, but then the gene change takes place. So then you have NF1 isolated to one body segment. And so we call that segmental NF. And then there's an incredibly rare phenomenon called gonadomosaicism. So this happens when only the gonad cells are involved. So this can be the sperm or the egg, but the cells that are going to be the sperm or the egg are the only cells in the body that have the gene change. So if you look at my example, let's see. I can't make it work. If you look at my example here, you can see that two parents who have no signs or symptoms of NF1 had two children who had NF1. And when you looked at the sperm of this individual, of the dad, you can see that he actually does have a variant in his sperm. And so we know that it was just the sex cells that are involved. Again, I want to stress that this is a very rare phenomenon, but I wanted to mention it because it is a possibility. So when it comes to genetic testing, the vast majority of individuals have had testing done via blood or saliva. And that's because most of the time we're dealing with that classic NF1 scenario. However, when we're dealing with a mildly presenting individual, we may need to utilize testing from a different tissue type. And so it would be affected cells like tumor or tissue or affected tissue. And then sometimes to help refine recurrence risk, we may also need to do testing on other tissue types like sperm. So to talk a little bit more, so we've been on the conversation of mosaicism. If someone is truly concerned that an individual is mosaic, it does require testing affected tissue to make that diagnosis. So the affected tissue for NF1 that we typically test is neurofibromas or cafe au lait spots. Now, these are very, what we call heterogeneous, but it just means that there's a mixed bag of cells that are a part of that tissue type. But it's specifically neural crest-derived cells that we have to test to see gene changes associated with NF1. And so we have to get those affected tissues and hone in on just those cells to do testing. For the cafe au lait spots, that special cell line is called melanocyte cells. For neurofibromas, that special cell line is called Schwann cells. And what's unique is that in blood, we only expect to see one gene change. But when we're talking about mosaicism in affected tissue, there's actually two gene changes that we expect to see in those tissues. One is the same in every single cafe au lait spot or every single neurofibroma. And it's because that's that one that was started from the embryological development. The second gene change just happened over time, but knocking out both copies of the NF1 gene caused that affected tissue to form. We also think about, in very mildly presenting individuals, the scenario of Legis Syndrome. So Legis Syndrome is characterized by just cafe au lait spots and freckling only, but it's caused by a totally different gene on chromosome 15. And so when you're looking at someone from the outside and all you see is cafe au lait spots and freckling, it can be difficult to make that distinguishment between NF1 and Legis Syndrome. And that's part of why, when all of the diagnostic criteria were revised last year, they made it specific that you have to have genetic testing to prove you have a SPRED1 gene change to make that diagnosis. Now, it's important to note that if you see a group of individuals and you put them in a room and they all have cafe au lait spots and freckling, the vast majority are still going to have NF1. Only about 2% of these individuals will have Legis syndrome, but it's an important distinction because you don't expect any of those neurofibromas to present in Legis syndrome. There can be other reasons that you can see a mildly presenting individual in NF1. And through our cohort of patients that have been tested at UAB, so about 8,000 individuals, we have been able to look at some of the clinical features that we've been told about when these individuals have come in for testing and been able to make some genotype, so the gene change, phenotype, clinical presentation, correlations. So there are some gene changes that we know if the gene change is located at a very specific address, we can potentially predict if this is going to be more severe or mildly presenting. The 992 address is one of those examples. If there's 3 specific letters that are deleted, then we know that these individuals do not present with any type of neurofibroma. There are no symptomatic optic pathway gliomas as well. And about 4% of them have low-grade asymptomatic brain tumors, but learning disabilities as well. But that's an important distinguishment to make. There's another location at the address 1809 where we also see no neurofibromas in these individuals, but what makes that location interesting is that there's a lot of overlap between NF1 and a different disorder called Noonan syndrome, where you can see cardiac defects, more specifically pulmonic stenosis. And so there's a higher incidence of these cardiac defects when there's a gene change at that specific location. So that's important for the providers to know, because then they know maybe we need to refer you to cardiology, and that's not something they may have done as a part of a routine genetics follow-up or referral process for NF. Then on the other end, we can have more severely presenting gene changes. So one of those is a deletion, and we'll go into more specifics about that soon. But we know that there are more neurofibromas that present at earlier ages when we have a total gene deletion of NF1. And also importantly, there's an increased risk for MPNSTs. There's also another address, 844 to 848. When we see gene changes in this area, we also see a higher incidence of plexiform neurofibromas that are visible from the outside and symptomatic spinal neurofibromas. So if you find one of these gene changes in a younger child, for example, who may only have cafe au lait spots at that time, that's helpful information for the provider to know, because they may need to change what they normally do for medical management of individuals with NF1. So we talked about total gene deletions, and I think just about everyone who has been involved in the NF1 world has heard that total gene deletions have a more severe presentation. But I just wanted to highlight that it's because it's not just the NF1 gene that gets deleted in these total gene deletions. There are some neighboring genes on both ends that have been associated with having a more severe phenotype. So there's 3 different types of total gene deletions, and it's because they each involve different neighbor genes. But the take-home point is that it's because of the inclusion of those neighboring genes that you have a more severe presentation. So as you can see, when you have especially a small child who's presenting with just a few findings, it can be a very difficult dilemma of how to move forward. Deciding if you should start with testing on blood versus another tissue type. Are we concerned about mosaicism versus classic NF1? And so astute providers will be putting all of this in their mind when they decide on a particular genetic testing approach for you. So now let's switch to the other end and talk a little bit more about the schwannoma-related disorders. The first is NF2. And so in the past, timestamp anytime before 2022, we used to think of NF2 as its own individual disorder caused by the NF2 gene. It's also autosomal dominant, and it's located on chromosome 22. And in the past, we have always known that it is also fully penetrant. So that means if you have that gene change, you're going to see some signs or symptoms. And the average age of onset was 22 years. We used to make that diagnosis of NF2 based off of the features. In the past, we thought that if you have vestibular schwannomas, it's NF2. And when you don't have vestibular schwannomas, it must be schwannomatosis. However, over the past, 1 or 2 decades, we realized that there's a lot more ambiguity in these 2 disorders than we thought. So there's a lot more overlap and a lot more individuals with schwannomatosis that actually have vestibular schwannomas than we originally realized. And so this year, there's been a move to make schwannomatosis an umbrella term to include all the genes that are associated with schwannoma predisposition development. So now that's NF2, SMARCB1, and LZTR1. So there's been a revision to the diagnostic criteria this year so that we can focus more on what gene change was identified so that we can make those diagnoses instead of simply focusing on which tumors are or are not present. Another important fact for NF2 is that there is a higher incidence of mosaicism for this condition as well. So we've talked about mosaicism through NF1. The process is the same for NF2 as well. It takes having both copies of the gene in the tumor to have a gene change in order to see those schwannomas present. So when we test more than one tumor and we see a common gene change from tumor to tumor, we know that we're dealing with mosaic NF2. If we see the gene change present in all of the blood cells as well, we know we're dealing with classic NF2. With schwannomatosis, things get a little bit more tricky. So first and foremost, there are 2 genes that have been associated with schwannomatosis. The first is SMARCB1. It was the first one identified in 2007, and then LZTR1 in 2014. And together, at best, they make up about 86% of the cases that we classify as having schwannomatosis. That's an important number, and we'll go back to that, but I want to make sure that you guys appreciate that it's not 100% of cases. So there is expectation that there may be other reasons for schwannomatosis out there. It's an autosomal dominant condition as well, but what's unique about SMARCB1 and LZTR1 related schwannomatosis is that there's incomplete penetrance. So we've talked about how NF2 and NF1 are fully penetrant, meaning if you have that gene change at some point in your lifetime, you're going to present with some sign or symptom. With schwannomatosis, that's not always the case. There have been a number of cases of individuals with LZTR1 related schwannomatosis, for example, who have lived to the age of 80 and has never presented with a symptomatic schwannoma. And so we call that incomplete penetrance because it's not enough to say that your parents don't have features, so they may not have it. It also means that if you have symptomatic schwannomatosis and you pass down that gene change to your child, we cannot predict if they will or will not have symptoms as well. The genetic testing in tumor of schwannomas associated with schwannomatosis is also a bit more complex. So instead of having gene changes in one gene, what we see is more than one gene being involved. So LZTR1 and SMARCB1 are also both on chromosome 22, but there's this 3-event process that we see in these tumors. The first is a gene change in either SMARCB1 or LZTR1. Then in the tumor, the other side is completely deleted, and it deletes the addresses where SMARCB1, LZTR1, and NF2 are all located. Back on the same side that you have your original hit in SMARCB1 or LZTR1, you see a unique gene change in NF2. And so seeing that same pattern in more than one tumor lets us know for sure that we're dealing with either SMARCB1 or LZTR1 related schwannomatosis. So in the past, you can see that we've had genetic testing as small bits as a part of the diagnostic criteria, but I show you this so that you appreciate that now with the revised criteria, genetic testing is a very huge component of getting that diagnosis, because from a clinical perspective, it's very difficult to really make that clinical diagnosis of schwannomatosis without having that genetic testing done. So I give this example to kind of put more context to it. If you have an individual who has had maybe 2 tumors and we test both of them, we can see in this particular case that there's a same gene change in LZTR1 in both tumors. That second event of a long arm of chromosome 22 loss is there. And then the NF2 gene change changes from tumor to tumor. And so because it does not stay the same, we know that LZTR1 is the driver of what's going on. Of course, we wish everything could be black and white and simple, and we can classify all of our cases into those bins, but sometimes that's not always the case. Sometimes we don't see a gene change in SMARCB1 and LZTR1. And part of that is because what we've already talked about, maybe there's another gene involved. But when we see the other 2 gene changes without that first one, we can at least say that it's chromosome 22q related schwannomatosis. When we don't see any gene changes at all in the schwannomas, then all we can say is that it's schwannomatosis not elsewhere specified. So that's now its own diagnosis. It means that I've had genetic testing, but they didn't see anything, and so it's not elsewhere specified. There have been a number of candidate genes that have been recently published that might also be attributing to schwannomatosis, but there's still a lot of work needed and research needed for these genes before they can be included as a part of diagnostic criteria. And then sometimes it's just not feasible to get genetic testing done on tumor. And so when we can't do that and we have to go off of clinical diagnosis alone, we are calling that schwannomatosis not otherwise specified. So the take-home message of all of this is that it's very complex. And an ideal scenario is that you want to be able to test all 3 genes before getting that diagnosis of NF2, SMARCB1, or LZTR1 related schwannomatosis. It's also ideal to test more than one tumor. So as a patient, your role in that can be that if you know you're having a tumor removed and you haven't had full genetic testing done, that you can advocate with your surgeons to make sure that that tumor is saved in a way that it can have genetic testing done later. And given all of these nuances, it's also very important to work with an experienced provider. So someone who has experience with schwannomatosis and neurofibromatosis type 2 as well. So genetic testing is playing a larger and larger role in determining diagnostic certainty these days. It helps also in ensuring accurate recurrence risks as well, because knowing if someone is mosaic versus classic lets us know if we're talking about an individual who has a 50% chance of passing down a gene change versus a lower risk. And so that empowers families to be able to make some of their decision-making for their families. And then many clinical trials and drug managements now also request that they see a gene change in one of these genes for further progression. And so sometimes genetic testing has played a role there. So because of that, there's been a need to make sure that the labs that are doing this testing can become more uniform in characterizing these gene changes as pathogenic versus benign or somewhere in between. What we've seen is that with the uncertainty and the use of just a more generic rule set, that there's been a need for a group to come in and kind of take some ownership of that. And so there is a group called the Clinical Genome Resource, or we call them ClinGen, and they are through the National Institute of Health. And their goal is to try to help in promoting the sharing of genetic and health data from patients, providers, laboratories, and researchers so that we can become more uniform in getting these gene changes classified appropriately. And so when doing that, they've created a database called Clinvar, and it's a place where lots of laboratories go in and they share any of the gene changes that they've identified, and then they put in how they classified it. So you can see in there are a lot of places where they're not consistent. One may call it likely pathogenic. One has called it a variant of uncertain significance, for example. But there's a need to kind of make a final decision on which lab is right. And so to do so, they have implemented a new process called variant curation expert panels. And the role of the expert panel is to get a group of individuals who have expertise in a particular gene to start with that initial set of rules that were made to be uniform for all genes and tailor them specifically to the genes in question. And so they bring together experts, clinicians, laboratory experts, and they are expected to share all of their data and resources. They then create a very specific subset of rules that get published so that all laboratories can use that information. And then they are making the final decision on gene changes when labs don't agree. So through the help of the CTF, we have now been able to create a variant curation expert panel for the neurofibromatosis and schwannomatosis related disorders. I hope you appreciate from this that this is an international group. It involves over 13 countries with individuals with expertise in everything from clinicians to laboratory scientists, researchers, and bioinformaticians who are all coming together with their levels of expertise to look at all of these 5 genes and try to make these rules for these conditions. So these are the individuals who are a part of our current group who's working on NF1. And so they've all divided into teams to try to focus on rules specifically associated with these 3 areas. And so their goal in the end is to try to create a rule set for these conditions that can be applied in determining the pathogenicity of the gene changes that are seen. Now, this won't be an overnight process. First of all, it's difficult to get 30-something people to agree on one thing, nonetheless a rule set. But the expected plan is that we'll have all 5 genes with rule sets available by 2025. And then we'll begin the process of going through all the variants from there as well. So this was a very short summary of genetics of NF, but I hope there was something in there for everyone. I want to make special acknowledgement to the Children's Tumor Foundation for all of their work that they've done in helping and funding the initiation of the Variant Curation Expert Panel. ClinGen for all of their work in creating a set of processes that we can use and follow to make this happen. And then the members of the VCEP who, in addition to the regular work that they're doing day to day, they're also taking the time to try to create these rules. Doctors Scott Plotkin and Elizabeth Castellano, as they're serving as the PIs for this study. And I also make mention here of Dr. Ludwin Messiaen, who is now retired, but has contributed a lot to the world of NF through her large data set of individuals and the ability to make these genotype-phenotype correlations. So thank you.
Well, thank you, Dr. Gomes, for that incredible introduction to NF genetics. My name is Kavita Sarin. I'm a dermatologist at Stanford University. And I just wanted to thank the meeting organizers for allowing us to present some of our research on NF1. And while our research today is going to focus mostly on NF1, the methods and fibromas, optic gliomas, scoliosis, are bony fractures. But most people develop subsets of these, and many develop mild cases and others more severe. And we call each of these a phenotype. So our manifestation of our NF1, for example, an optic glioma, is called a phenotype. So even though we all have NF1 mutations, the manifestation is slightly different. And our study is trying to understand why this is. 3 years ago, we undertook a collaborative study with CTF to try to understand if we could determine patterns in manifestations of NF1. So we received de-identified data on about 2,000 individuals with NF1 who have reported, self-reported phenotypes, including their skin findings, plexiform neurofibromas, optic gliomas, and fractures. And if you look broadly at the findings in this group, it's pretty much what we would expect. Many people have skin findings, like 98% of this cohort had cafe au lait macules, many axillary freckling, about 30% had plexiform neurofibromas, and so on. But what we wanted to understand was there a pattern in manifestation? Were these just randomly distributed, or was there actually a pattern that was occurring across this cohort? And this is, many of you are in the CTF registry, so thanks to you, we were able to do this type of study. So we did something called hierarchical clustering, where we took 18 different self-reported phenotypes of NF1, and we looked to see if they clustered together. For example, if you had one phenotype, were you more likely to have another phenotype? And this is called a cluster analysis. And what this means here is that, for example, if you have bowing of your bones, you're more likely to have osteoporosis. You're also more likely to have optic gliomas. Axillary freckling. So if axillary freckling is present, there's more likely to be groin freckling, cafe au lait macules, and CNF. So these are sort of our skin cluster. Scoliosis and fractures are more likely to cluster together, along with plexiform neurofibromas and spinal neurofibromas. And attention deficit is more likely to correlate with itch and pain, as well as female and family history. And so there are patterns. It's not a completely random roll of the dice. There's actually patterns of manifestations or phenotypes in NF1. We also look to see if individuals are more likely to be similar to each other. So are our manifestations in NF1 more likely to be similar? And this complicated plot is called a Tisney plot. And basically what we identified was 6 different clusters of people, when we cluster people who are more likely to be similar. So if everyone just randomly got an attribute of NF1, our phenotype, you would imagine that all these clusters, 1, 2, 3, 4, 5, 6, would be just randomly distributed all across. They'd be all jumbled together. But what we found was that there's a group of people who are more similar. We call them cluster 1, more similar cluster 2, 3, 4, and 5. And when we looked at the phenotypes that caused these individual clusters, 1 was more mild, 2 was more freckling predominant, 3 had more neurofibromas, 4 had more skeletal, recurrent fractures and skeletal findings, and then 5 and 6 had more neural findings. Why is this? Why do we have patterns of manifestations of our NF1? And we hypothesize this is all due to, or significantly influenced by our genetics. And Dr. Gomes explains really nicely, and I won't go over it again, how we've identified a number of mutations either in the NF1 gene or deletions that are associated with unique phenotypic manifestations of NF1. And a lot of this has been thanks to an incredible database at University of Alabama. And Dr. Gomes was heavily involved in this work as well. But those genetic mutations only encompass less than 10% of the phenotypic variability seen in NF1. And our hypothesis is that this phenotypic variation actually occurs through mutations and variations outside of the NF1 gene. And why is this? Well, this was a beautiful study done by Easton et al. In 1993, so almost 20 years ago, that looked at 48 families with NF1. And so for him, this is an example of a family. So say grandfather has the NF1 mutation. His daughter and son both have the NF1 mutation. And then his grandchildren, through his daughter, also have the NF1 mutation. Well, they noticed a very interesting finding. Among identical twins, the probability or correlation of neurofibroma numbers, cutaneous neurofibroma numbers, were practically identical. They were all very similar. Among parent-child combinations, it was still correlated. So the amount of neurofibromas like a parent had was more similar to that of a child, but significantly less, so a correlation of 0.19. If they looked at siblings that were non-identical, so didn't share identical DNA, there was a correlation, but again, much weaker, 0.15 as compared to 1, which is a perfect correlation. And then if they looked at grandparent-child, so the genetics are still similar, but they're getting further apart, they found the correlation was even lower, 0.05. And then if you're starting to look at cousins, or what we call third-degree relatives, the correlation was much lower. So what does this suggest? Well, all of these individuals have the exact same NF1 mutation. Why is their correlation not perfectly correlated, right? And this is because most likely there's genetics that run in the family outside of the NF1 locus that influence their manifestation. But what those genetics are, we still don't know. And it's not just genetics, it's a little more complicated than that, right? There's other things, perhaps hormones or environmental exposures, other experiences that could influence the development and trajectory of our NF1. One way we go about identifying genetics is called a genome-wide association study. And this study is actually a very simple study. The way it works is we take individuals that we call cases with a condition. So for example, let's say we wanted to study and identify the genetics associated with optic gliomas. Well, we would take a bunch of people with optic gliomas, they're called cases, and we would compare their genetics to people without optic gliomas or controls. And we would do a variety of methods of genome sequencing. It could be whole genome sequencing or array-based genotyping. And we would look at whether or not there were any mutations or variants that were enriched in the patients with optic gliomas compared to those without. And this has been done to identify the genetics of over 4,000 different GWAS studies have been done to identify genetics related to many different conditions. And this is the approach that we're interested in taking for NF1. And there actually have been some GWAS studies already conducted in NF1. There was a GWAS study for cafe au lait macules that found a possible risk variant, but that did not replicate. And this occurred in 112 participants. There's another study, not a traditional GWAS, but that tried to look at monozygotic twin and parent pairs, so identical twins and parrots, for copy number variations, but also did not find anything of significance. And this is likely because there were just not enough participants in these studies. These genome-wide association studies usually take thousands or sometimes tens of thousands of participants to be able to identify these genetic findings. And that is the goal of our research and why we're here talking to you today, and while we're launching this massive effort to create a genetic biobank to understand the genetic modifiers that influence our course of NF1. And this database is going to be crowdsourced because the only way to do this is to have many people participate, and you can imagine how difficult it is for a rare condition like NF1. And so what we're collecting is clinical data, genetic data, and photographs from 1,150 individuals with NF1 over the age of 40. And it's in the United States as well as international locations. And the initial study will be to see if we can conduct a genome-wide association study to evaluate the genetic variants that influence the trajectory of cutaneous neurofibromas. As you can imagine, this is a massive effort to crowdsource the number of participants we need to be able to make genetic findings for cutaneous neurofibromas, but for any NF1-related phenotype. And so Akshika Patel, who's outside our booth here, is our lead clinical research coordinator coordinating this event. We're also collaborating with Dr. Romo and Dr. Blakely, a huge number of NF support groups and clinics who have helped promote this study and encourage participants in this study, and this study is funded by NTAP. And what I thought I would do is actually just show a video on how you could get involved. So if we could go ahead and play this video.
Have you been diagnosed with neurofibromatosis type 1 or NF1 and have one or more cutaneous neurofibromas? Do you want to help improve the lives of others living with NF1? Researchers from Stanford and Johns Hopkins are studying the genes of people 40 and older living with NF1. They want to better understand why some people are more likely to develop certain disorders like cutaneous neurofibromas. You could have an impact on the future of NF1 care by participating in this research study. You may learn about genetic markers that could impact your health and could be passed down to your children or possibly shared by other family members. Participation is easy. No need to schedule or travel to doctor's appointments. And when you're done, you'll get a $25 Amazon gift card. To participate in the study, you'll need to complete a survey, send us photographs of your skin, and provide a sample of your saliva for genetic testing. You can do all of this in about an hour from the comfort and privacy of your own home. The study is open to residents of the US and select international sites, such as Canada and the EU. Just visit our website at www.nf1genetics.org, complete an eligibility checklist, and sign the consent form to get started. With your help, we may develop new therapies and provide more personalized care, improving the lives of millions of NF1 patients around the world.
So, we'd encourage everyone who's watching virtually online or here in person to get involved. And if you're here in person, I don't know how to go to the next slide, if you can help me with that. There's a second on the video. If we'd encourage everyone who's here in person to come out to the booth, and you can get everything done in about 30 minutes right outside of the booth in this room. Could you help me progress the slide? It stopped working. Thank you. Our partnership is with Nebula Genomics, so everyone who participates will get whole genome sequencing. And then if they would like to, at the end of 3 months, when they get their data back, they'll actually get access to a portal where you can explore your own whole genome DNA beyond that of NF1. You can look at a variety of traits that might be associated or risk factors that you may have based off of your DNA. Next slide, please. So recruitment is ongoing. We've collected 545 enrolled patients who've completed surveys. We have thousands of photographs from 345 individuals and are getting DNA from whole genome sequencing data back from 280 individuals. Next slide. Great. We're predominantly female and predominantly white, so encouraging everyone to participate. And the number of neurofibromas our participants have ranged from very few all the way up to more than 500. So we've got great representation. We've taken some initial looks at this data set, and what we can find is that the number of neurofibromas that our participants have are associated with their itch scale that they're reporting. So our number of neurofibromas does seem to correlate with itch. We also looked to see if ultraviolet light might induce neurofibroma formation. So we looked at the zones of where people live and whether or not high ultraviolet light zones might be associated with higher numbers of neurofibromas, and could find no correlation. So we built this database to crowdsource genetic and clinical data for NF1. We have robust enrollment, but we're only halfway there, so we need a lot more people to join. And we need to continue to forge connections. So please reach out to us if you have any connections or want to help in this study. And if you're not interested in genetics, I'd still encourage you to think heavily about the study. And so I thought I'd like to just share a learning from alopecia areata. So alopecia areata is an autoimmune disease where our own immune cells attack our hair follicles. And 10 years ago, a GWAS study came out. It was the first ever genetic study of alopecia areata done in 1,000 patients, and identified this pathway called a JAK-STAT pathway that was involved, and mutations were occurring in this pathway in patients with alopecia areata. And that led to the development of JAK-STAT inhibitors for the treatment of alopecia areata. And the first JAK-STAT inhibitor, baricitinib, was just approved last month for alopecia areata. And you can see this tremendous effect this inhibitor has. So this is pre-treatment and post-treatment of alopecia areata. You can see significant hair regrowth. And this pathway all came from a GWAS study. And so I encourage you to participate. I think we're hoping that we can build a database that will provide answers not only for cutaneous neurofibromas, but help understand the genetics of a variety of phenotypic manifestations of NF1. Thank you very much.
Okay, I think we have some time for a couple of questions. If we have any questions in the audience, I can bring you the microphone. Just raise your hand. for either of our speakers? Great.
I'm an NF mom. It's never dawned on me that anybody would be interested in my genetics. I'm always looking at the research studies for my son. Is that something like he's spontaneous, so I assumed my genetics were of no value to this, but are they? Is that something I should change my mind about?
Okay, yeah, so that's a great question. And so I think the answer to that is twofold. So first, in terms of looking at the NF1 gene, it's unlikely that any variants or any that you have in your NF1 gene contributes or makes any impact for your son, because that's a spontaneous mutation. However, based off of some of the data I just showed you, our other variants, we don't know which ones yet, but our other variants that we may have that are common variants in the population could influence someone's NF1 trajectory. And that's what we're trying to understand now. And so, we can understand that primarily by studying individuals with NF1, but it is true that it's very likely that your other common variants that you carry can influence his trajectory outside of the NF1 gene. Excellent. Another question? Yes. Okay.
Thank you to both speakers. Really, really interesting. I'm an NF1 mom, but also have a background in genetics, so this topic is of particular interest. So thank you for bringing that to light, and thank you for the work that you're doing. This question is actually more for CTF in terms of the registry and kind of like, do we have a biobank so that when researchers like these women are looking to do genetic studies, that they can pull from a database rather than individually recruiting for each study? And if not, is that something that we as a community could look to do so that we could do these studies in terms of the recruitment faster, and then maybe more of them as well?
Yes, thank you.
So yeah, that's an excellent question. So we had a biobank that we started in 2013, I guess, in Frederick. And when we saw that there was a big pickup on especially cutaneous neurofibromas, we in fact transferred the biobank now to Indianapolis, to Indiana University, where there is somebody dedicated whose day job it is to manage biobanks. So the short answer is yes, there is a biobank. And then in Hopkins, there is a nice biobank as well. So it's, again, not duplicating efforts that are happening, right? But there are, yeah.
I can also just say that the plan for this genetic study is, because the recruitment is, it's going to take years and it's difficult, is to then deposit this in a source that researchers who want to study genetics associated with any area of interest of theirs could then access the genetic data as well as the clinical survey data in this database with applied entry, because it's obviously patient information. But and so I think that having a resource like this, just at least on the genetic side, with whole genome sequencing data tied to clinical reported data, can be used by a variety of researchers in the future.
Excellent. We have a question from the Hula app that I think it would be really helpful for everybody to hear the answer. So somebody says, excellent presentation, Alicia. It might be helpful to comment on which services are effective for NF genetic testing. There was a question in the app about 23andMe and these types of testing, so if you could comment on that.
So that's actually a great question. Not every genetics service provider is the same. You want to make sure that you're working with a clinical diagnostic lab. So these are labs like, I'm just using examples, this is not a full set. There are commercial labs like Invitae, GeneDx, Ambry. There are academic labs like the lab at UAB, and also internationally available academic labs. These labs follow a very specific certification process to make sure that they are uniformly getting the same results every time. And also, they have a vested interest in making sure that their testing is as thorough as possible. When we're talking about labs like 23andMe, or what we call direct-to-consumer labs, these laboratories aren't necessarily looking at the whole gene. So their goal is not to necessarily be thorough. They are most of the time catching hotspots if they look at NF1 at all. So most of them aren't looking at NF1. So if you've had any type of direct to consumer testing, meaning that you were able to spit in a kit without a provider being involved in any way. That is not true clinical diagnostic testing. You do want a provider involved that has determined that the test that you're doing really does give you as close to that greater than 95% detection as we can get. It's a great question.
Yes, thank you so much. I'm sure there are so many more questions, but in the interest of time, we are going to move on. Please be sure to put questions in Huva. I know, for example, that Dr. Gomes has already been in there responding, so please be sure to take advantage of that. Oh, yes. Oh, and I just wanted to mention that Dr. Sarin's lab is outside recruiting for their study, but please be sure to visit the exhibit hall because we also have some folks recruiting for an NF1 in sleep study. And then in the Huva app, we've sent some information about the inflection study and one out of Yale. So please be sure to visit. I know Dr. Booley is outside for her sleep study, so be sure to check all of that out while you're here. Thank you so much to both of you.
Thank you.